Hello class 9th students, this is Praful Sharma, biology lecturer, TUP School Amit. This lecture is the summary of chapter 5 NCERT book The Fundamental Unit of Life chapter. The chapter includes definition of cell, cell size and shape, types of cell, cell structure, cell wall and its structure, nucleus, its content, cytoplasm, cell organelles like endoplasmic reticulum, Golgi apparatus, ribosome, mitochondria, lysosome, centriole, plastid, vacuole, and cell division. The first heading is cell. Cell is the structural and functional unit of life. It is termed as structural unit because it provides structure to our body, and it is considered as a functional unit of life as all the functions of body take place at cell level. Cells were first discovered by Robert Hooke in 1665. He observed the cells in a cork slice with the help of a primitive microscope. Leeuwenhoek, with the improved microscope, discovered the free living cell in pond water for the first time. It was Robert Brown in 1831 who discovered the nucleus in the cell. The cell theory was expanded and we can say was propounded by two biologists, Schleiden in 1838 and Swan in 1839. It was further expanded by Rudolf Virchow in 1855 by suggesting that all cells arise from pre-existing cells. With the discovery of electron microscope in 1940, it was possible to observe and understand the complex structure and the cell and its various organelles. The invention of magnifying lenses led to the discovery of microscopic world. It is now known that a single cell may constitute a whole organism, as in amoeba, chlamydomonas, paramecium, and bacteria. These organisms are called unicellular organisms. On the other hand, many cells group together in a single body and assume different functions in it to form various body parts in multicellular organisms, such as some fungi, plants, and animals. Can we find out names of some more unicellular organisms? Every multicellular organism has come from a single cell. How? Cells divide to produce cells of their own kind. All cells thus come from pre-existing cells. Some organisms can also have cells of different kinds. Look at the following pictures. It depicts some cells from the human body. The shape and size of cells are related to the specific function they perform. Some cells like amoeba have changing shapes. In some cases, the cell shape could be more or less fixed and peculiar for a particular type of cell. For example, nerve cells have a typical shape. Each living cell has the capacity to perform certain basic functions that are characteristic of all living forms. How does a living cell perform these basic functions? We know that there is a division of labor in multicellular organisms such as human beings. This means that different parts of the human body perform different functions. The human body has a heart to pump blood, a stomach to digest food, and so on. Similarly, division of labor is also seen within a single cell. In fact, each such cell has got certain specific components within it known as cell organelles. Each kind of cell organelle performs a specific function, such as making new material in the cell, clearing up the waste material from the cell, and so on. A cell is able to live and perform all its functions because of these organelles. These organelles together constitute the basic unit called the cell. It is interesting that all cells are found to have the same organelles no matter what their function is or what organism they are found in. Organisms with a prokaryotic cell do not contain a true nucleus. They are primitive and incomplete cells. Prokaryotes are always unicellular organisms. You can see that in the diagram, like bacteria, blue-green algae. These are unicellular organisms. They are prokaryotic as well because they do not contain any membrane enclosed organelle, or we can say a typical cell. A eukaryotic cell are the cells in which the true nucleus is present. They are advanced and complete cell. They include both unicellular as well as multicellular organism except the blue-green algae and bacteria. So a typical well-complex cell is a eukaryotic cell. The prokaryotes and eukaryotic cells have multiple differences in between them. Say for example, a prokaryotic cell is too tiny in compare of a eukaryotic cell. The nucleus is not found in prokaryotic cell, but it is present in eukaryotic cell. A prokaryotic cell has smaller ribosomes, where a eukaryotic cell contains obviously larger ribosomes in terms of size. Let's move on to the next heading, that is the structure of a cell. We saw that the cell has special components that we call as cell organelles. How is a cell organized? If we study a cell under a microscope, we would come across three features in almost every cell: cell wall and cell membrane, nucleus and cytoplasm. All activities inside the cell and interactions of the cell with its environment are possible due to these features. Let us see how. Plasma membrane or cell membrane. This is the outermost covering of the cell that separates the content of the cell from its external environment. The plasma membrane allows or permits the entry and exit of some materials in and out of the cell. It also prevents movement of some other materials. The cell membrane, therefore, is called a selectively permeable membrane. How does the movement of substance takes place into the cell? How do substances move out of the cell? Some substances like carbon dioxide or oxygen can move across the cell membrane by a process called diffusion. Water also obeys the law of diffusion. The movement of water molecules through such a selectively permeable membrane is called osmosis. The movement of water across the plasma membrane is also affected by the amount of substance dissolved in water. Thus, osmosis is the passage of water from a region of high water concentration through a selectively permeable membrane to a region of low water concentration till equilibrium is reached. Cell wall. Plant cells, in addition to the plasma membrane, have another rigid outer covering called the cell wall. The cell wall lies outside the plasma membrane. The plant cell wall is mainly composed of cellulose. Cellulose is a complex substance and provides structural strength to plants. Cell walls permit the cells of plants, fungi, and bacteria to withstand very dilute external media without bursting. It determines the shape and rigidity of plant cell. It protects the plasma membrane. It prevents desiccation or dryness in the cell, and it also helps in transport of various substances in and out of the cell. Here comes the controlling center of all cell organelles, that is nucleus. The nucleus has a double layered covering called nuclear membrane or nuclear envelope. The nuclear membrane has pores which allow the transfer of material from inside the nucleus to its outside, that is, to the cytoplasm. The nucleus contains chromosomes which are visible as rod-shaped structures only when the cell is about to divide. Chromosome contains information for inheritance of characters from parents to the next generation in the form of DNA. Chromosomes are composed of DNA and protein. DNA molecule contain the information necessary for formation and organizing cells. Functional segment of DNA are called genes. In a cell which is not dividing, this DNA is present as a part of chromatin material. Chromatin material is visible as entangled mass of thread-like structure. Whenever the cell is about to divide, the chromatin material gets organized into chromosomes. The nucleus plays a central role in cellular reproduction, the process by which a single cell divides and forms two new cells. It also plays a crucial part, along with the environment, in determining the way the cell will develop and what form it will exhibit at maturity. In some organisms like bacteria, the nuclear region of the cell may be poorly defined due to the absence of nuclear membrane. Prokaryotic cell also lack most of the other cytoplasmic organelles present in eukaryotic cell, just as of nucleus. Many of the functions of such organelles are also performed by poorly organized part of cytoplasm. The large region of each cell enclosed by the cell membrane is the cytoplasm. This region takes up very little stain. It is called the cytoplasm. The cytoplasm is the fluid content inside the plasma membrane. It also contains many specialized cell organelles. Each of these organelles performs a specific function for the cell. Cell organelles are enclosed by membranes. In prokaryotes, besides the absence of defined nuclear region, the membrane-bound cell organelles are also absent. On the other hand, the eukaryotic cell have nuclear membrane as well as membrane-enclosed organelles. The significance of membranes can be illustrated with an example of viruses. Viruses lack any membranes and hence do not show characteristic of life until they enter a living body uses its cell machinery to multiply. What do we mean by diffusion? The movement of molecules from a region of their high concentration to a region of their lower concentration is known as diffusion. You can see that in this diagram where a small drop of dye, when is inserted or dropped into water, the particles of dye, they expand everywhere in the water due to diffusion. On the other hand, osmosis is the movement of water across a semi-permeable membrane, the membrane which allows movement of solvent particles especially. Osmosis is a selective process since the membrane doesn't allow all molecules to pass through it. There are three types of solution on the basis of their concentration. A solution is said to be isotonic when its concentration is same as of cell shape. If it is said to be hypotonic, if the solution's concentration is lesser than the concentration of cell shape, and it will be called as hypertonic when the concentration is higher in compare of cell shape. Let's start with the cell organelles. Every cell has a membrane around it to keep its own contents separate from the external environment. We have talked about the nucleus. Some important examples of cell organelles which we will discuss now are endoplasmic reticulum, Golgi apparatus, etc. Let's start with the Golgi apparatus first. The Golgi apparatus, first described by Camillo Golgi, consists of a system of membrane-bound vesicles, flattened sac, arranged approximately parallel to each other in stacks called cisterns. These membranes often have connections with the membranes of endoplasmic reticulum and therefore constitute another portion of a complex cellular membrane system. The material synthesized near the endoplasmic reticulum is packaged and dispatched to various targets inside and outside the cell through the Golgi apparatus. Its function include the storage, modification and packaging of products in vesicles. In some cases, complex sugars may be made from simple sugars in Golgi apparatus. The Golgi apparatus is also involved in the formation of lysosomes. The endoplasmic reticulum is a large network of membrane-bound tubes and sheets. It looks like long tubules or round or bag-like vesicles. The ER membrane is similar in structure to the plasma membrane. There are two types of ER, that is rough endoplasmic reticulum RER and smooth endoplasmic reticulum SER. RER looks rough under a microscope because it has particles called ribosomes attached to its surface. The ribosomes which are present in all active cells are the site of protein manufacture. The manufactured proteins are then sent to various places in the cell depending on need using the ER. The SER helps in the manufacture of fat molecules or lipids important for cell function. Some of these proteins and lipids help in building the cell membrane. This process is known as membrane biogenesis. Some other proteins and lipids function as enzyme and hormones. Although the endoplasmic reticulum varies greatly in appearance in different cells, it always forms a network system. Thus, the function of ER is to serve as channels for the transport of materials, especially proteins, between various regions of the cytoplasm and nucleus. The ER also functions as a cytoplasmic framework, providing a surface for some of the biochemical activities of the cell. In the liver cell of the group of animal called vertebrates, SER plays a crucial role in detoxifying many poisons and drugs. Ribosomes are extremely small, dense and spherical bodies which occur freely in the cytosol, or they may remain attached to the endoplasmic reticulum. These are made up of ribonucleic acid, that is RNA, and protein. They are of two types especially, that is 70S ribosome, which is found in prokaryotic cell, and 80S ribosome, which is mostly found in eukaryotic cells. The function of ribosomes is to synthesize proteins. The next cell organelle is mitochondria. Mitochondria are known as the powerhouse of the cell. Mitochondria have two membrane coverings. The outer membrane is porous, while the inner membrane is deeply folded. These folds increase surface area for ATP generating chemical reactions. The energy required for various chemical activities needed for life is released by mitochondria in the form of ATP, that is adenosine triphosphate molecules. ATP is known as the energy currency of the cell. The body uses energy stored in ATP for making new chemical compounds and for mechanical work too. Mitochondria are strange organelles in the sense that they have their own DNA and ribosomes. Therefore, mitochondria are able to make some of their own proteins. If we talk about their function, they are the site of cellular respiration, hence provide energy for various activities of living cells. As they help in producing ATP, they are also known as powerhouse of the cell. The next cell organelle we need to discuss is centrosome or centrioles. It is found only in eukaryotic animal cells. It is not bounded by any membrane but consists of centrioles. These are long, hollow, cylindrical structures arranged at right angle to each other and made up of microtubules. This organelle helps in cell division, as well as it also helps in formation of cilia and flagella, the locomotory organs in many organisms. Here comes plastids. Plastids are present only in plant cells. There are two types of plastids: chromoplast, which are colored, and leucoplast, which are colourless. Chromoplast contains the pigment chlorophyll. At that time it is of green color and we call it chloroplast. Chlorophyll are responsible for sunlight capturing. Chloroplast also contains various yellow or orange pigments in addition to chlorophyll. Leucoplasts are primarily organelles in which materials such as starch, oils and proteins are stored. The internal organization of chloroplast consists of numerous membrane layers embedded in a material called stroma. These are similar to mitochondria in external structure. Like the mitochondria, plastids also have their own DNA and ribosomes. If we talk about their function, chloroplast trap solar energy and in this way help in photosynthesis. Chromoplast, because of their various color, they makes the flower attractive so that they can attract the insect for pollination. Leucoplast helps in storage of various food products, say for example starch, proteins and fats. The next cell organelle is lysosome. Structurally, lysosomes are membrane bound sac filled with digestive enzyme structures. These enzymes are made by rough endoplasmic reticulum. Lysosomes are a kind of waste disposal system of the cell. These help to keep the cell clean by digesting any foreign material as well as worn out cell organelles. Foreign materials entering the cell such as bacteria or food as well as old organelles end up in the lysosomes, which break complex substances into simpler substances. Lysosomes are able to do this because they contain powerful digestive enzymes capable of breaking down all organic material. During the disturbance in cellular metabolism, for example, when the cell gets damaged, lysosomes may burst and the enzymes digest their own cell. Therefore, lysosomes are also known as suicide bags of the cell. Vacuoles are storage sac for solid or liquid content. Vacuoles are small sized in animal cells, while plant cells have very large vacuoles. The vacuole of some plant cells may occupy 90% part of the cell volume. In plant cells, vacuoles are full of cell shape and provide turgidity and rigidity to the cell. Many substances of importance in the life of a plant cell are stored in a vacuole. These include amino acids, sugars, various organic acids, and some proteins. In single-celled organisms like amoeba, the food vacuole contains the food items that the amoeba has consumed. In some unicellular organisms, specialized vacuoles also plays important roles in expelling excess water and some waste from the cell. Each cell thus acquires its structure and ability to function because of the organization of its membrane and organelles in specific ways. The cell thus has a basic structural organization. This helps the cell to perform functions like respiration, obtaining nutrition, and clearing of waste material or forming new proteins. Thus, the cell is the fundamental structural unit of living organisms. It is also the basic functional unit of life. As we already discussed that new cells are formed in organisms in order to grow, to replace old, dead and injured cells, and to form gametes required for reproduction. The process by which new cells are made is called cell division. There are two main types of cell division, mitosis and meiosis. The process of cell division by which most of the cell divide for growth is called mitosis. In this process, each cell called mother cell divides to form two identical daughter cells. The daughter cells have the same number of chromosomes as mother cell. It helps in growth and repair of tissues in organisms. Specific cells of reproductive organs or tissues in animals and plants divide to form gametes, which after fertilization give rise to offspring. They divide by a different process called meiosis, which involves two consecutive divisions. When a cell divides by meiosis, it produces 4 new cells instead of just 2. The new cells only have half the number of chromosomes than that of the mother cells. Can you think as to why the chromosome number has reduced to half in daughter cells? Just to maintain the number of chromosome equal as of a normal cell in a offspring. Here we are done with this chapter 5, the fundamental unit of life.